In this series of factorial design, we will look into the different steps that are needed to formulate and how to achieve an optimum formulation and how to use this to formulate different dosage forms. So we'll follow a step-by-step method to end up with, in this case, as an example, we're going to use 3 to the power of 2 design. The problem we're having is that if you follow this kind of approach, trial and error, this is what we call a traditional approach. So this is what you often do. You change one variable at a time. In many cases, this follows random experiments, and there is no way you can cover the same area that you can do using factorial design. So that could lead to waste of materials, time, and resources. And because it's random and it's not systematic, may completely miss the optimal region that we should really aim for. This doesn't follow any mathematical model for predictions, and it cannot measure any factor interactions. And we will look into this more in detail later on. So our solution is the factorial design, and this is a strategic way where we do the experiment in very systematic way. So every combination of factor levels is tested, and we aim for a coverage to ensure that we reach an optimum region. It generates a mathematical model or an equation, and it will measure the effect, main effect, and interactions. And altogether, it will enable us to reach a confident prediction for any kind of combination of variables. The general formula that is used in factorial design is shown here and is expressed as x to the power of n. That gives us the total number of experiments that we need to do. X here is the number of levels per factor. That means low, medium, or low, medium, high. So usually either 2 levels, low and high, or low, medium, and high is three levels. This is the most common, and we're going to follow this. N is the number of factors or variables. Basically, what variables you want to change to test. And in many cases, we were going to choose 2 factors. And in this case, so we have 3 levels, means low, medium, high, to the power of 2 variables or factors, and that will give us 9 experiments in total. If we imagine if you want to have 3 factors, then you'll end up with 27. So that means you'll have increased the number of experiments. So we need to be also economical in terms of the cost and time. And then the idea, the whole thing is to plan minimum experiments for maximum information. Watch this animation carefully. It shows how we build a 3 squared grid. So we start with our first factor on one axis. So say for example we are making tablets. So on one axis will be the binder concentration. We mark out the 3 levels we mentioned before, which is low, medium, and high. Then we add our second factor on the other axis, and let's say that factor is the compression force. And then again the same 3 levels, low, medium, and high. Now we draw the grid lines. So where they interact, we get our 9 experimental points. Each point represents a unique combination. So that's 9 combinations in total. Each one becomes an experiment we need to run. This grid is the foundation of factorial design. It ensures complete, as you can see here, complete systematic coverage of the experimental space. So you might be wondering why we use 3 levels, why not just 2, for example, high and low. It's a good question to ask or think about. The answer is to do the shape of the relationship between your variables. with just two levels, you can only draw a straight line between your points, but you can, that way you can detect linear relationships. So when doubling the input, that doubles the output. But let's assume, for example, this is not a linear, and this is often the case in pharmaceutical formulations, and that results in a curved relationship. So, for example, maybe increasing the binder from 2 to 4% gives you a big improvement, but going from 4 to 6 gives you hardly any extra benefit. So that means there is a curved relationship. So with the three levels, low, medium, and high, you can detect this curvature. The middle point tells you whether this relationship uh, is straight or there is some kind of curvature. So although that means more experiments, basically 9 instead of 4, but the extra information is invaluable here. So you get much more accurate model for your system. So throughout this course, I will be using an example to make everything clear and just more easy to understand. And the example I will use often is a tablet formulation. And the reason for that, because tablets are the most common dosage forms commonly used in pharmacy. They seem simple, but getting the formulation right requires careful optimization. So in our experiment, we are going to study how tablet hardness is affected by two factors. So tablet hardness means how easy they break. So we will use two factors. The first one is the binder concentration. That is the ingredients that holds the tablet together. And the second factor is the compression force. And that tells us how hard we need to press the powder in the tablet press to get the tablet. So our goal is to find the combination of those two factors that gives us the tablet with the right hardness. So it shouldn't be too soft, and likewise, shouldn't, on the other hand, shouldn't be too hard, because otherwise it will not dissolve well. Before we go further, let's understand what makes a good tablet. So there are several quality properties we need to measure. Uh, Often the hardness of the tablet, and this is measured in newtons. It tells us how resistant the tablet is to crushing. So if it's too soft, it will break during handling or packaging. Or on the other hand, if it's too hard, it might not dissolve at all. So clearly here that's not good. The other parameter or the other measure we use is the friability. This is measured as percentage, and it tells you how resistant the tablet to chipping and abrasion. So the weight loss must be less than 1%, and this is actually based on, so you need to check that in the pharmacopoeia as well for these values. The other thing is the disintegration time, and that is measured in minutes, and it tells you how quickly the tablet breaks apart in liquid. So when you put it, how long does it take to break? The dissolution, once it's disintegrated, we measure the dissolution, and this is measured as a percentage, often is measured as a percentage, and tells us how much drug actually dissolves over time. In our example, we'll focus mainly on hardness. So we'll look at one factor just for simplicity. But when you do optimization, you need to take more than one, ideally maybe three. And that is the hardness in this example is the response variable. And we'll come on this later on to explain that in more detail. So this is the Y value in our equation.